right, so just to let you know, because we've had, some folks have to miss class for various reasons, I'm recording the lectures and they're posted on Brightspace just in the, so like all the chapter 1 videos are going to be posted in the chapter 1 folder. So we're going to finish chapter 1 today and start chapter 2. So today's will be in the chapter 2 folder. All right, so we're going to whip through the last part of chapter 1 by just talking about the different types of mutations. So remember, the information is stored in the DNA that eventually winds up in the production of proteins. And if there's a change in the DNA due to some stress factor on the cell that causes an error in copying, that can result in a change in the identity of the monomers that get put into the protein. But it doesn't necessarily have to, okay? So it could result in a change that's drastic like this, and this type of mutation would harm function or eliminate the function of that protein. But you could, for example, instead of, if you had a change instead of a glutamic acid, an aspartic acid, which is, it's essentially the same functional group that's going to be on the side. It's just the chain is 1 carbon shorter. So you still have a carboxylic acid group there, okay? And so that would be a more conservative mutation that might affect function, but it wouldn't eliminate it entirely, unlike this particular example, okay? All right. Okay, so we can have a variety of different types of mutations, and again, this is how different functions evolve, but it's also how sometimes bad things can happen, okay? So we've got the original sequence represented up above, and if we have a change in only one position, that's called a point mutation, okay? So this is only one position. Now there are different stages, okay, right, at which mutations can occur. They can occur when you are, when a cell is dividing, right? So any daughter cell would have that mutation. They could occur when the DNA is being transcribed into RNA. So that would be kind of a transient mutation, right? And they can be in what are called germline cells that would be inherited. Okay, so these type of mutations would result in inherited genetic diseases. Or they could be in, like a cell of, a somatic cell that's, let's say, on your elbow, for example, that it just so happens to, over the course of your lifetime, that mutation develops. But again, it's not going to be in one of those cells that gets passed on to your offspring, so it would be not inherited, okay? And so it's just limited to you. So an example of this would be something like a cancer that would develop over the course of your lifetime that is related to, let's talk about maybe skin cancer, right? So it's not in, your egg or your sperm cells that get transmitted into the offspring, but let's say it's, again, on your arm, and those cells are exposed to the sun and they get mutations, and when they are dividing, then a cancerous tumor develops, and that only affects you. It's not going to affect any offspring you may have. Okay, so there's two types, okay, of broad classes of cells where you can get mutation. Okay. So talking about, putting things in an evolutionary context, okay, the random mutations that we see that do get passed on to offspring, okay, can result in beneficial properties that get selected for by natural selection, okay? So, excuse me. If you have mutations, again, they can occur in the areas where the gene is regulated, or they can occur in the coding region. Okay, so it's a difference of what is the product being made versus how frequently and under what conditions it's made. Okay, so these flanking regions, okay, that don't code for the final protein do actually have a lot of control about how that protein functions in the cell, because they control its presence and level and things like that. Okay. So basically, every time a mutation occurs, the individual that has that mutation either has a survival and reproductive advantage, or they don't. Okay, and it gets passed on or not. And so over time, fewer mutations are going to occur in the coding region. Okay, so this would be what gets made, than in the flanking regions, because over time you kind of optimize the function of that protein, and once it's optimized, it's not going to be, there are going to be fewer mutations that actually result in a survival advantage. Okay. All right. Okay, so in addition to having like a base pair change, okay, we also can have genetic rearrangements occurring in cells. Okay, so we can get genes being copied so that you now have more than one copy of a gene. Okay, and that can have a lot of differing results. Okay, so it's not always, more is better. Sometimes more is worse, and sometimes more is different, and sometimes more is just more. Okay, so if we look, let's say, we've got a gene that gets duplicated, okay, and this is where more is better, right? Where having 2 copies of the gene is better than 1, because it kind of doubles your ability to produce the material when needed. Or we could have 1 copy acquire a mutation, and now we have a new function, right? So the cell acquires new abilities. And you can think of this, when we talk about protein function, we'll talk about hemoglobin and myoglobin and multi-subunit proteins. When we talk about kinetics, we'll talk about enzymes that behave differently in different tissues because the subunit composition differs. Okay, so you can imagine, okay, in this case, where we get a new function from the copied gene, it's got a slight mutation that maybe makes it do the chemistry faster and produce product faster. And that might be good for a liver cell, okay? But in a heart cell, you maybe don't want it to produce product faster, but you want to grab on to the reactant faster, okay? And so, those mutations can also optimize the use of the gene for different organ types. Okay. All right. And then, the other thing that can happen is over time we get rid of one of the copies because it doesn't provide any advantages. We don't have any new functions, and it's not needed. So that's what happens with gene duplications. Okay. So if we have something that acquires a new function, okay, this here is called paralog, okay. All right. Okay, so that sort of ends our tour of the central dogma of biology and talking about the major classes of molecules we're going to study. So what we're going to do now is we're going to move into chapter 2, where we've already talked a bit about intermolecular forces in lab. So today we're going to be talking about energy, okay, and the laws of thermodynamics. Okay, whoops. Okay, so chapter 2 again is going to review concepts of energy, so endothermic and exothermic reactions. We're going to, remind ourselves about the different types of components of energy that contribute to what we call free energies, which is whether or not reactions occur. Okay? So we'll talk about the different ways we can calculate free energies or use information about free energies to tell us about what's controlling the behavior of a system. Okay. And those things that control the behavior of a system in biochemistry boil down to intermolecular forces and the impact of the pH of the environment, which again we're working with in lab. Okay. And then we will talk about membranes and how they control the environment to a certain extent. All right. So if we're talking about energy, we have to start with the sun, right? And the sun is where all energy is going to come from, okay? And the way biological processes you access and use this energy, okay, is going to be through the molecules, okay, that control our metabolism. Okay, so organisms that can directly use energy from the sun, okay, use molecules that can change their conformation in when they are exposed to radiation, and a series of electron transfer reactions. Okay, so ultimately we go from solar energy to electron transfer to chemical energy, which is how cells can store energy for when they want to use it. Okay. And so there's a lot of reactions that, even though they may be thermodynamically favored, okay, don't happen because kinetically they are slow without a catalyst. Okay, so that's pretty much the story of biochemistry, is reactions are controlled through catalysts and through using energy to manipulate concentrations. Okay, so we're going to talk about the relationship between concentration and free energy. And then when we talk about enzymes, we're going to talk about the impact of catalysts. Okay. All right. So when we talk about energy in biological systems, we call it bioenergetics. So how energy moves from one source in one place to another and how it gets used and conserved. So we have to, biologically speaking, we have to start with photosynthetic organisms, whether they're plants or photosynthetic microorganisms. Okay, so they can take solar energy and convert it into chemical energy, right, through the reactions of photosynthesis. So there's kind of two phases. They're called the light reactions, and then these are often called the dark reactions or the Calvin cycle. So these are the light reactions, and those are dependent on incoming radiation or electromagnetic radiation. And then these are often called the dark reactions or the Calvin cycle. So the light reactions, okay, take the energy from the sun, okay, so they take the electromagnetic radiation, and what they do is they convert it into ATP and reduced NADPH, okay? So we've got chemical energy stored in ATP, and we have a reduced cofactor, okay? And so a big theme in, cellular energetics is that you have a reaction where some energy is harnessed, okay, and it's used to reduce a molecule. And that enables us to take those electrons, okay, and that molecule can diffuse somewhere else. And then if we can oxidize that molecule, okay, and move that electron, we can get energy out of that process, okay. So the way we move electrons, okay, is not through copper wires in cells, but it's through cofactors, okay, and so NADPH is one of them, okay. So what happens is the ATP and the NADPH go into this second series of reactions called the Calvin cycle, okay. So the light reactions require light and water, okay, and they produce oxygen. This is how plants wind up giving us oxygen, a net of oxygen, and ATP and NADPH. And then the part of your plants that's going to take in CO2 is using, okay, the products of the light reactions to make carbohydrates. Okay, so the sugars that the plant cell uses as food, that animals and organisms that consume the plants are going to be taking in. And so we take this sugar and then we convert it to energy through through glycolysis and the citric acid cycle and oxidative phosphorylation. Okay, so glycolysis, citric acid cycle, and oxidative phosphorylation to convert sugar to energy. So the energy comes in from the sun, goes out through sugar, and then we convert it into energy by breaking down the sugar and oxidizing it, okay? Yeah. All right. So why do cells need energy to survive? And the main reason is that there are a lot of processes and chemical reactions that are needed to sustain life that cannot occur without an input of energy. Okay, so what's one thing you need to do if you're an organism that needs to, so going back to like middle school biology, what was like the definition of a living thing versus a non-living thing? What can living things do that non-living things can't? So, yeah, they can maintain homeostasis, so they can maintain like a specific body temperature, right? So that's one thing that we have to do, and that's going to require energy, okay? Because we have to be able to sometimes, under some circumstances, produce heat, right? What's another thing living things can reproduce, okay? So if you're going to make a copy of yourself, okay, it doesn't just magically happen, right? The cells have to make copies of their molecules, and every one of those reactions, okay, is going to take energy. All right, so the main kind of types of work that cells have to do, okay, break down into sort of three categories. Okay, so the first is osmotic, and that has to do with controlling where the water is. Okay, so controlling where water is and where water isn't. Okay, so if you look at the example that's shown up here, okay, this has to do with, maintaining the pressure in capillaries. So this is a small capillary, okay? And if we've got, if we have a situation, okay, where the capillary pressure, okay, is greater than the pressure in the blood, okay, then what happens is fluid is going to exit the capillary, okay, and we call that filtration. So, all right. And if we have a case where the blood and the capillary are kind of equal in their pressures, okay, we're not going to have any movement, okay, so we'll just have no net movement. Things will be the same. The blood's going to flow beyond the capillary, not enter the capillary, okay. But if we have a case where the pressure in the capillary is lower than, you know, outside in the blood vessel, okay, then what's going to happen is we're going to get liquid moving back into the capillary to get us back to the point where we have an equilibrium of pressure. Okay. And so sometimes I can't make my slide go back to small. There we go. All right. So sometimes the process of maintaining an osmotic balance occurs without extra energy input because of the varying concentrations of solutes across a membrane. But sometimes things can't cross membranes without help. Okay, and so sometimes you have to use energy from ATP to kind of push something across a membrane. All right. So the next type of work that there is to do, okay, is chemical work. Okay. All right. So we are making molecules and degrading molecules all of the time in cells, right? And that takes energy. Okay. So there's two types of pathways in metabolism, okay? And so the one that builds up molecules, okay, are called anabolic. So these build molecules. And catabolic pathways break down molecules, okay, to harvest energy. And these build molecules, and they use energy. Now, the one thing that is deceptive about this picture, okay, is that any individual reaction in, say, a catabolic pathway might use energy. So when we say in catabolic pathways, energy is released, okay, that means the end product of a pathway. An individual reaction, okay, may require ATP to make that reaction go forward. Okay? And so if you've ever gone through the steps of glycolysis in a biology class, you'll know that the first half of glycolysis is what we call the energy investment phase, and the second half is the energy harvesting phase. So you have to put in a couple of ATP, but you get twice as much out, so you net more ATP than went in. So the pathway, okay, produces energy, but individual reactions may require energy. Okay, so the important thing to remember, okay, is this is in terms of the end result of a pathway. Individual results may vary, as they say. So reactions along the way may require energy. But at the end, anabolic pathways use energy and catabolic have a net gain. Okay. So those asterisk statements are the net of that type of pathway, not any individual reaction. Okay. All right, and then the last type is mechanical, and that's muscle contraction in animals. When we talk about protein function, we will talk about the actin-myosin cycle, okay? And so this is probably the most kind of conceptually easiest for us to observe, because we know what it feels like when you move your arms or legs a lot, right? You feel like you're using energy. All right. Okay. Oops. So different types of energy. Again, we've got our solar energy and our photosynthesis. So when we talk about those three types of work that we talked about, this is just giving you sort of a microscopic example of each. So the osmotic work that we talked about before has to do with, again, not everything can move through a membrane just by diffusion alone. Okay, and so sometimes you need help to allow, let's say this is a polar molecule or a polar ion that can't get through the nonpolar membrane, so it has to go through a channel that goes through the membrane, okay, and that channel can require ATP hydrolysis to kind of open and close to allow things through. Okay. So that's an example of the type of osmotic work that might need energy. Right? And then chemical work, again, along our pathways, some of those reactions might need ATP, and some of them won't. Some of them can just go on concentration. And then mechanical work, that's our muscle contraction. All right, so in order to be able to do work, right, we have to be, as Gabby said, we have to be in a state of homeostasis. So what we mean when we talk about homeostasis is that we've got an organism that's in an ordered state. We're not just blobs of molecules floating around. There's an organizational structure to it, okay? And that includes conditions like temperature, but it also includes where molecules are and how much of them are in different places. Okay, so remember our square brackets represents concentration. So this is where, whoops, and how much of a particular molecule is. Okay, so does anybody know what the equilibrium state for a living thing is? So if a living thing is allows all of its chemical reactions to get to equilibrium without any extra expending of energy, what would you be? Dead. Yes. All right. So in order to not be dead, okay, we have to have some reactions that are not in their equilibrium conditions. So remember that equilibrium, okay? Equilibrium is a reversible chemical reactions happy place, okay? So in terms of the ratio of reactants to products where things are not going to change, they're going to stay net the same, okay? And so every chemical reaction that is reversible has a different spot, okay, a different particular ratio of reactants and products that is optimal. Okay, so if we're at equilibrium, we're not going to be able to maintain homeostasis. And so if we look at the rabbit, he's sitting here in the classroom. That's 20 degrees Celsius, roughly room temperature. But internal temperature, okay, that both humans and rabbits maintain is 37 degrees Celsius, right? And so that heat has to come from somewhere, okay? And if you look at the rabbit, he's got ears, he's got eyes, he's got whiskers, he's got a nose, he's got a cute little puffy tail. So there's an organizational structure there. So that organizational structure is kind of a low entropy state, okay? And so all of those things are going to require energy, okay? All right. All right. Okay, so redox reactions. Redox reactions, again, are the way that we store energy, or that we move energy into ways that we can store it, and the way that we get energy out of some stored molecules. Okay. All right, so redox reactions in biology, we have series of them where your electron carriers, okay, are all arranged in a way that their reduction potentials, okay, are going to be ordered so that it's easy for one molecule to pass the electron on to the other, okay. So we're going to get a transfer of electrons from one molecule to another in a sequential fashion, so there's an order to it, okay, and that order, again, is in order of reduction potentials, okay. And they're going to be used, anytime we have some chemical work done, and energy is, needed or made, okay. And this figure shows you something that is super common in biological reactions, which is called coupling, okay. So coupling of reactions, okay, means that I have one chemical reaction, okay, that maybe releases energy, and I have another one who has, if we talk about endothermic and exothermic reactions in Gen Chem, right? We talk about endothermic reactions like needing an energy input to get them to go forward, right? Maybe you have to heat it, maybe you have to, do something else to get it to go. Now, if you're a biological organism, we're going to talk about the impact of heat on the different types of molecules that we study, okay? And what we're going to find is that, proteins and DNA, if you heat them high enough, okay, they're going to lose their structures, and their structures are required for their specific function. So, biological organisms cannot make unfavorable reactions go by just always increasing the heat, right? So they have to be able to make them go in another way. So what they do is they couple exothermic and endothermic reactions so that the energy released by one can make the other reaction go forward, okay? So one releases and one uses the energy. And so that's why the organizational structure is necessary. Okay. All right, so I told you we like to use a simplified, or not necessarily a simplified definition of oxidation and reduction, but because we see repetitive patterns, we can recognize when oxidation or reduction is happening when we see, so if we have reduction, okay, that if we see a gain of electrons, so maybe a net charge decreases, or we see more hydrogens, okay, or we see a decrease in the number of bonds to oxygen, okay? So we don't have to be calculating oxidation numbers if we just look at our molecules before and after and we say, did we start with something charged and now it's neutral? Did we start with something that gained a hydrogen, not a proton, but a hydrogen with its electron, okay? Or did we see, did we lose a bond to oxygen? Did we have, a carbonyl and now it's a hydroxyl group, okay? So that tells us, one, we lost a bond to oxygen, and two, we gained a hydrogen, right? And then oxidation is just kind of the inverse of that, right? We get the loss of electrons, a decrease in the number of hydrogens, or an increase in the number of oxygens, okay? So, if you look at this example here, okay, we have water, okay, that is becoming O2, okay? So in that case, okay, we are losing hydrogens, so our system, is being oxidized, right? So water oxidized to oxygen gas. In the next, so when that happens, okay, so it's being oxidized, and whenever we have an oxidation reaction, remember, electrons have to go somewhere, right? So something has to be reduced. Those electrons that are being lost from the water have to go somewhere, right? And in this case, okay, what happens is we have a quinone cofactor, okay, that gets reduced. Okay, so this cofactor gets reduced, okay? And in this case, this is such a favorable process that we also have enough energy released to make ATP, okay? All right, so, but now I have my reduced cofactor, okay? And that's going to be able to pass electrons, these 4 electrons, to a cofactor NADP+. Okay, so these are reactions in photosynthesis, okay, that are going to be able to pass the electrons now to the NADP. And so we get that reduced. All right. So whenever we talk about energetics in a physical chemical context, we have to kind of define like what we're talking about. If we want to talk about is energy coming in or going out, you have to think about, well, am I just looking in the room? Am I looking in the building? Am I looking at the globe as a whole? So we define systems versus surroundings. Okay, so we have isolated systems that are, they cannot exchange energy or matter with their surroundings. We have closed systems where they can exchange energy but not matter, okay? And we have open systems where, everything's a go. You can exchange energy and matter with your system. So biological organisms kind of live, right, in an open environment, but because we have areas of organisms and cells that are kind of sequestered away and maybe control the exchange of matter, we can have some examples of closed systems within what we're looking at, okay? And a system just means, what's the region of space you're talking about? Are you talking about the room, or are you talking about the building? And then the surroundings is everything that's not included in the system you're talking about, okay? So I can talk about the system of the cell, or I can talk about the system of the organism. All right. So our laws of thermodynamics. Excuse me. So what's called the zeroth law is the one that has to do with thermal equilibrium and the way that heat is going to travel. Okay, and the ones that are most important to us in biochemistry are the first and second laws, which are conservation of energy. Okay, so energy that comes from somewhere has to go somewhere else. Okay, and so changes in energy are related to amounts of work done. And then the second law is the definition of entropy that says, in all spontaneous processes, the entropy of the universe increases. So we'll talk about a more sophisticated definition of entropy than maybe you dealt with in Gen Chem. All right, and then absolute zero. Okay, again, in biology and when we study biological systems, we really don't talk about absolute zero much because we need to have our molecules moving to not be in that permanent state of equilibrium. Although we do use the absolute temperature scale when we calculate free energies and things like that. Okay, so our first law, again, energy can either be created or destroyed. So again, energy has to be converted from one form to another, okay? And if work is done, okay, then, energy gets lost, but it gets transferred to the thing that we're doing work on. So we've got chemical energy, which in biological systems, we store it in ATP, mostly ATP. It's stored in a couple other ways. Okay. And then mechanical energy, again, organisms that move, and there are plants that move too. I don't know if you've ever seen the pictures of the walking trees, the walking forest, but there are these trees that they move their roots. They have these tall roots. It's pretty cool. But so this is, for an example of this is, muscle contraction. But even if you're a microorganism, you can move if you don't have muscles, right? Certain types of bacteria have cilia and flagella, and they move, and that requires chemical energy, uses lots of ATP, okay? All right, and then energy itself, whether, it's chemical or mechanical, okay, it can be of two types, potential energy or kinetic. So this is energy of motion, and potential energy is energy that is not actively doing anything at the moment, but could, right? All right. So, again, because we don't have systems that are 100% efficient, okay, we often lose energy when it moves from one place to another. Okay, so if you've ever driven a car that has like a tire pressure sensor, and especially on like fall days when it's cold at night, all right, and the gas in the tire kind of contracts because of the lower temperature, you get in the car, the low pressure sensor may be on, but as you drive the car, okay, that will often go away because as the tire, rolls along the road, there's some friction there, okay, and And some of the energy that goes into moving the tire and going across the pavement gets converted to heat that gets transmitted through to the gas inside the tire. It heats up a little bit, and now your car's sensor is like, okay, we have enough air in the tires, right? All right, so if we talk about heat energy being lost, okay, we call that enthalpy, okay? So, oops. So this is work, and this is change in energy, and this is our enthalpy. Okay. So in chemistry, we talk about enthalpy mostly in terms of chemical bonds. Okay, the amount of energy that gets released when a chemical bond or a favorable intermolecular interaction, intermolecular force gets made, right? Bless you. Okay, so exothermic reactions are reactions that release heat, okay? And those are going to have a negative delta H. So the things that are important for you to remember in terms of enthalpy, okay, is, okay, delta H is negative for exothermic reactions, and we typically think of these things as favorable processes, okay? Or favored by enthalpy, okay? So entropy and enthalpy can both be favored, or one can be favored, the other can be unfavorable in a given situation. So if your delta H is negative, that's something that will be spontaneous, okay? If, the entropy is not considered, okay? So endothermic reactions are ones that absorb heat, okay? And so they're going to be taking in heat, so that enthalpy change is going to be positive, okay? And so a positive enthalpy change is going to be unfavorable in terms of what the enthalpy change contributes to a reaction going forward. Okay. So, examples of exothermic and endothermic chemical reactions are ones that occur in those instant hot packs and cold packs, right? One of them gets hot because it releases heat to its environment. The other gets cold because it sucks heat away from its environment. All right, so this is a good time. So we will talk about entropy in class on Wednesday, and then we'll do some free energy calculations. A note, if you have a laptop, bring it to lab with you tomorrow, okay? So we're going to be doing experimental stuff, but also some computational stuff. You can bring a tablet. So the computer stuff we're going to be doing is going to be using WebMO, which is you just need a browser, and we're also going to be making some standard curve graphs. So there actually is a really good app that you can download on a tablet called Data Analysis. My undergrad thesis advisor wrote that, and I actually never realized that he was the author of the app until I started using it in class, and I went to go update it, and I'm like, oh, Evan Kantrowitz, Dr. K. It's like, I never knew. I'm like, no wonder it's so useful, because it's all the stuff we used to do. But anyway, all right.
Thank you.
See ya.
Just for clarification, so we have chapter 1 homework and chapter 2